In this video, we'll be discussing about Helicobacter pylori infection signaling. To understand this process, let's first look at the cell membrane. The cell membrane acts as a protective barrier regulating what enters and exits the cell. It is the first point of contact for pathogens like Helicobacter pylori. Now here on the cell membrane, we can see we have the Type IV Secretion System. This complex molecular machinery is used by Helicobacter pylori to interact with the host cell. It functions like a syringe, allowing the bacterium to inject its virulence factors directly into the host cell. Inside the cell, we have the nucleus. Now to start the signaling pathway, we see Helicobacter pylori comes in and binds with the Type IV Secretion System, from where it injects CagA into the cell as shown in the diagram. Then we get the CagA phosphorylation, which then drives the activation of SHP2 molecules, followed by the activation of JAK state pathway. We see the unregulated activation of this JAK state pathway leads to cancer promotion. On the other hand, CagA molecule also activates ERK pathway, which also leads to cell proliferation, thus promoting cancer. Secondly, we have the secretion of VACA molecule by pylori. VACA molecule binds to specific receptors on the host cell membrane, such as lipid rafts or receptor protein tyrosine phosphatase beta receptors. Then we see this VACA molecule enters the cell where it targets the mitochondria to release cytochrome C, which leads to formation of ROS, thus driving the DNA damage. Furthermore, we see this Helicobacter pylori can modulate our immune system effectively. As shown in this diagram, we see Helicobacter pylori attaches to the host cell surface using two key adhesin molecules, BabA and SabA. These proteins facilitate surface recognition by binding to specific receptors on the host cell membrane. This attachment is a crucial first step as it allows Helicobacter pylori to initiate signaling cascades that modulate the host immune system. Following this attachment, two major signaling pathways are activated: TLR pathway and NOD pathway. Let's first see the TLR pathway. The interaction of Helicobacter pylori components with toll-like receptors like 2, 4, 5 triggers the TLR pathway. This leads to activation of MyD88 molecule, an adapter protein crucial for downstream signaling. MyD88 molecule then activates NF-κB pathway. This NF-κB then translocates to the nucleus and induces the expression of pro-inflammatory cytokines, contributing to the immune response. Simultaneously, NOD1 and NOD2 receptors inside the host cell recognize peptidoglycan fragments from Helicobacter pylori. This activates RIP2 molecule, which in turn stimulates the MAP pathway. The MAP pathway or MAP cascade leads to activation of AP1, that's activator protein 1, which regulates gene expression involved in inflammation. Both these pathways, TLR pathway and NOD pathway, converge to produce pro-inflammatory cytokines, including IL-1 beta, IL-8 and TNF-alpha. These cytokines drives the recruitment of immune cells, which leads to tissue damage. This continuous cycle of inflammation and tissue damage results in chronic inflammation, ultimately leading to gastritis or cancer. So this is the basic mechanism of Helicobacter pylori infection signaling. I hope you like the video. If you like it, give it a thumbs up. Do consider supporting my work on Patreon or YouTube and make sure to subscribe to the channel. Thanks.